Every time that a cell copies its DNA, it has to add the complements to millions or even billions of letters, and it really has to do it perfectly or it could develop problems. There are enzymes that are responsible for double checking this copying process. To some degree, the DNA polymerases do this, and there are also enzymes known as nucleases that go back over the DNA and read it and make sure it's been copied properly. However, mistakes do happen in spite of this. We call mistakes in the DNA mutations. Mutations are random changes to the DNA sequence, but they are oftentimes permanent changes to that sequence. Here, for instance, there was a guanine in the original sequence and it's been changed to an adenine. And here's another mutation. There was a thymine in this sequence and now that's been changed to a guanine as well. Mutations can cause problems for cells because they change the information that's actually stored in the DNA. So they can make changes to the proteins and RNAs that are built based on that information. By far the most common cause of a mutation are simply errors in the copying process. The machinery of the cell that carries out DNA replication makes a mistake and sticks the wrong letter in at the wrong position. Remember that any time a cell goes through DNA replication, it has to carefully, even perfectly copy millions or even billions of letters at a time. Now, even given that, cells don't make mistakes very often. But when you think about the number of times they have to do this in an average day, you can get quite a few mutations. Think about this. Bone marrow cells make red blood cells by dividing. They can divide as many as 150 billion times a day to make a new ounce of blood. So within those 150 billion times, and think about the 8 billion letters they're copying each time, there's a lot of potential for making mistakes. Another potential cause for mutations is environmental damage. Typically, this is exposure of cells to things like UV light, as in this poor gentleman, or really nasty chemicals, things we call mutagenic chemicals. Exposure to these things can directly disrupt the DNA, or it can affect the machinery that works with the DNA so that they make more mistakes. This is the reason that exposure to UV light, that tanning, can cause cancers. That UV light can affect the DNA of a skin cell. That skin cell can start to grow out of control, and it can cause a tumor, a cancer. There are many different types of mutations, and they can affect DNA in different ways. There are several different ways to classify mutations as well. One way to classify mutations is based on the amount of DNA that they affect. If a single set of letters is affected, we call that a point mutation. Now, even though point mutations only affect one pair of nucleotides, they can be really powerful, and we'll see an example of that later on. Another way to classify mutations is based on how they affect the DNA. For instance, in this mutation, this pair of guanine and cytosine has been swapped out for an adenine and its complement thymine. Because the letters have changed, we call that a substitution. Mutations known as insertions and deletions also affect the nucleotide sequence. In an insertion, a nucleotide pair or pairs is inserted into the sequence, so letters are added to the sequence. Here, for instance, we have the sequence CTT, and you can see that an insertion mutation has occurred here because now the sequence is CATT. A deletion is the opposite of an insertion, so it's the removal of a pair or pairs of nucleotides. Inversions and translocations tend to be larger scale events, so they tend to happen with more than just a single pair of nucleotides, although they can occur with a single pair as well. In an inversion, a section of DNA is cut out, flipped over, and glued back into the sequence. And because the two sides of DNA are not identical to each other, this is going to change the sequence of both strands. In a translocation, a section of DNA will be cut out, carried somewhere else in the cell, and glued into another sequence. And that, of course, can change the sequence as well. Another way to classify mutations is based on the type of cells that they affect. Somatic mutations occur in what we call somatic cells. These are body cells that are not reproductive. So somatic mutations can occur anywhere in the body other than the testes or the ovaries. So let's say that a beam of UV light comes down from the sun and it strikes this skin cell right here on this guy's side and causes a mutation that causes a cancer or a growth. Now that's bad news for him. He's going to have to deal with that cancerous growth. But the good news about somatic mutations is that they aren't passed on to the next generation. Because it happened in a little skin cell right there on his side, and that skin cell is not going to be passed on to his kids, he's not going to pass that cancerous gene on to his kids. Germline mutations occur in cells we call gametes. Gametes are sex cells, so gametes include the egg cell and or sperm cells. Now, germline mutations can be a little bit more insidious because remember that a fertilized egg is going to divide and divide and divide, and it's going to build the body of the offspring. So a mutation that occurs in an egg cell, for instance, is going to be passed on to the next generation. And that mutation will occur in every single cell of that offspring's body. So germline mutations can be passed down from mother to child or from father to child, and can cause problems in the offspring. Incidentally, if you've ever gone to the dentist and had to get x-rays, you might notice that they make you wear this giant lead apron. And you might have also noticed that that lead apron does go down over the lap area. The reason they're making you wear that apron is to prevent this type of mutation. They're going to zap your body with x-ray radiation. That can potentially cause mutations, and they want to make sure that your internal organs, including your reproductive organs, are protected from this. So how often do these crazy changes occur? Well, scientists estimate that about 1 in 1,000 nucleotides are incorrectly copied every single time the cell goes through DNA replication. And when you consider the number of divisions that our cells go through during a day, and when you consider the lifespan of a human being, these somatic mutations can really build up over time. Scientists also estimate that every single human being is born on average with 60 germline mutations. So those are mutations that occurred in the sperm cells and the egg cells that conceived each of us. And so in a sense, we are all mutants. Not in the cool X-Men kind of way, but in more of the realistic scientific kind of way. Incidentally, I should mention that not all mutations are bad. We often think of mutations as being associated with diseases like cancer, but most mutations are neutral and don't affect the body at all, or mutations can even be beneficial. This is what drives evolution. For instance, a person might carry a mutation that changes an enzyme so that they can eat a new food source and have access to that. So not all mutations are bad. Finally, we need to discuss how DNA is stored inside your cells. Just to remind you, your DNA is split into these long sections called chromosomes. So a chromosome is just a long piece of DNA. If I were to look inside a nucleus of one of your body cells, I would find 46 of these chromosomes. 46 is the magic number for human beings. Different organisms have different numbers of chromosomes. Now on those chromosomes, I'm going to find the individual recipes for making specific proteins or RNAs. We call those specific recipes genes. So a chromosome really consists of a bunch of genes that are strung together with some other little segments of DNA in between, which we'll talk about in a later chapter. Consider this. If I took the DNA from a nucleus of one of your cells, if I took all 46 chromosomes and stretched them out end to end, I would have a molecule that was nearly 6 feet long. And yet all 6 feet of this DNA can be crammed inside a nucleus that is far too small to see with the naked eye. How is that even possible? Well, it's all in how the DNA is stored. I want you to think of DNA as being kind of like yarn. If you've ever worked with yarn for a crafting project or a knitting project, you know it can really kind of be a mess. You don't want it to just be stored randomly or willy-nilly. You want your yarn to stay organized so it's easy to work with. So with yarn, you take the yarn and you ball it up. You either ball it into a skein or a nice tight little ball. DNA does something kind of similar. Here is linear DNA. It's like a long piece of yarn. That long piece of yarn will be carefully wrapped around these proteins called histones. So it gets wrapped around and around and around these guys, and that helps ball them up. Collectively, a histone and the DNA that's wrapped around it is called a unit called a nucleosome. That fiber will then be twisted up on itself a little bit more to form something called chromatin. Chromatin is the working form of DNA. Most of the time during its life cycle, DNA is going to be in the chromatin form. It's loose enough for enzymes to get in and work with it, but it's not so loose that it's disorganized or going to get damaged.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Hopefully this chapter has provided you with a little bit more information about DNA, about how it's put together and how the cell uses it. We're going to use this information quite a bit more in future chapters, so keep it in mind. And remember, never ask a science nerd a question that you don't want a really thorough answer to. Bring your questions to class and we'll work on them. See you next time.